So we'll now look at case 3. Case 3 looks at a 5-year-old patient who was brought into the pediatrician. The mother notices some symptoms in her child. The symptoms that she notices are persistent thirst and frequent urination. So right off, these symptoms to me showed that the patient had diabetes. So to test for diabetes, blood samples are taken for insulin measurements, both at time 0, and then the patient is given a glucose loading test, and 1 hour after this test, the blood insulin levels are taken again. So the results of this test are shown here in this table. So the baseline insulin levels of the patient were around 0.8, which is quite low because a control patient should be around 6. After 1 hour of waiting after the glucose loading test, the insulin levels were still very low. They appeared to be about 5, where a control patient should be about 40. So these insulin tests again suggest that the patient has diabetes, specifically type 1 diabetes, and is not able to produce insulin. So the biochemical basis of this is the patient is not able to produce insulin, which we know is a very important hormone in the regulation of glucose uptake. So without insulin, GLUT4 transporters cannot transport glucose into certain tissues, specifically into skeletal muscle and adipose cells. So as these tissues do not receive glucose, the patient will often be very weak and tired. So the predictions of the patient's fasted blood glucose levels, due to the diabetes, the patient will likely have very high blood glucose levels in the fasted state. This is due to the hypoglycemia present at the beginning of the fasted state, which will cause the liver to release an excess amount of glucose, causing the blood sugar to be very high, and this will continue to persist throughout the entire fasted state, so that the blood levels are kept very high because many tissues are not receiving the glucose that they need. So two regulatory enzymes that are likely to be active in this patient, I looked at 2 in the liver specifically, because the liver will transport glucose in through GLUT2 transporters, which are insulin independent. So these in the liver regulatory enzymes are continually active even without insulin. So the first enzyme that I looked at was glucokinase. So in the liver, as glucose is continually transported in, glucokinase will phosphorylate this glucose to make glucose 6-phosphate. This glucose 6-phosphate can then be used for different things in the cell. This takes us to our next regulatory enzyme, which is glycogen synthase. Glycogen synthase will convert this glucose 1-phosphate to glycogen. This is done through the process of glycogen synthesis, and again, glycogen synthase is the main regulatory enzyme that does this process. So in the fed state, glycogen synthase is dephosphorylated and it is caused to be active. So these two enzymes, again, as they are present in the liver, which is able to transport glucose in, even with insulin deficiency, will cause them both to be active in this patient. So that concludes our case study for Unit 2, and thank you for listening.